Good evening and heartiest welcome to all the participants in today's invited talk by Professor KARTIK SHANKER on finding Philautus, the evolution and biogeographic of peninsular Indian herpetofauna organized by BioLogic. We are all here today to be a part of this adventurous journey to find Philautus. The title itself is so fascinating that it creates interest about the unknown world of herpetofauna. A few days back, we celebrated Save the Frogs Day to raise awareness about these amazing amphibians. I, with my classmates, participated in the event. And today, with a new energy and excitement, I hope we will all learn something from our honorable speaker. We have received an overwhelming response from all over India showing interest in this unique topic. The participants are from 20 different states spanning from Kashmir to Kanyakumari and from western region to northeast part of India. And even countries like Bolivia, Nepal and Nigeria are participating. So now I would like to invite Dr. Sandeep Pal, Assistant Professor Zoology, Barrackpur Rashtraguru Surendranath College, Professor to, and the founder and admin of BioLogic to deliver the welcome note and introduce today's guest speaker. Sir, please.
Um, Thank you, Ambika. So on behalf of BioLogic, I welcome all the students and other participants once again. Today is a very special day for me as I am sharing the same platform with the person I admire a lot. It is indeed a dream comes true moment for me. Now it's time to introduce today's invited speaker. Kartik Shankar was inspired to a career in ecology by an ancient reptile, a sea turtle, that crawled ashore late one night in Madras. As faculty at the Center for Ecological Sciences, Indian Institute of Science, Bangalore, he works on the ecology and evolution of frogs, reptiles, birds, plants, and marine fauna. He has set up long-term monitoring programs for sea turtles across the country, including olive ridleys in Orissa and leatherback turtles in the Andaman and Nicobar Islands. He has also served as the president of the International Sea Turtle Society and regional vice chair of the IUCN SSC Marine Turtle Specialist Group. He is also a founding trustee of Dakshin Foundation, which works largely with coastal communities on natural resource conservation and management, and a founding editor of the magazine Current Conservation. He is the author of the book From Soup to Superstar, a historical account of sea turtle conservation in India, and has written several children's stories such as Turtle Story, The Adventures of Philautus Frog, Moonlight in the Sea, and Lori's Magical Mystery. So we all are waiting to hear from Professor Kartik Shankar. Before starting today's session, this is to inform you all that the whole event comprises of 45 minutes of talk followed by 15 minutes of question answer session. If you have any relevant queries or questions, please ask at the end of the talk, only in the live chat box. Now, the stage is all yours, Professor.
Thank you, Sandeep, for that very generous introduction. And Sandeep and Ambika for organizing this. Gives me great pleasure to be here and share some of my work with you. Let me see if I can get this going pretty.
Okay, sir, sure.
Okay. Are you able to see my
Yes,
screen
sir.
now?
Yes,
Yeah.
yes,
And on full screen, right?
yes,
Great. All right. Shall I get started?
yes, sir, sure.
Okay. So today I'm going to be talking a little bit about evolution, how we study evolution more broadly, and then some of our work on the evolution and biogeography of herpetofauna in peninsular India. And for those evolutionary biology students over here. Some of the first part of what I talk about might be a little basic, but I just thought I would present as broad a background as possible. I also thought I would mention that, again, for students of ecology and zoology out there, our lab does actually quite a few different things. As Sandeep very kindly mentioned, what I'm going to be talking about today focuses on our work on diversity, distribution, and biogeography, and you'll hear much more about that. But we also work extensively on mixed species foraging, groups in both birds and reef fish. We study the community ecology and behavior of such groups, and I've spent several decades now studying marine turtles, which has led to more recent work on marine biology, on coral reefs and reef fish. And I just wanted there's more information on my website should you want to know about that. The part of our work in the lab that I'm going to be talking about today relates to patterns of diversity. And if you look at large scale global patterns of diversity, you can see that there are very clear gradients, and these gradients manifest themselves for whichever taxonomic group you're looking for. So if you look at amphibians, you see this very, very high diversity in the tropics and in mountain regions, and you see this across South America, Africa, Asia, as well as Australia. You see a similar pattern for plants. Again, high species richness in tropics, much lower in the temperate regions. You see a pattern actually very similar to frogs for fish, and that leads us to the question: what causes diversity patterns? Now, on the one hand, of course, there is this broad pattern that uh, the tropics have more diversity than temperate regions, but also within the tropics, some areas have greater diversity than others. And we can ask that question: why do some of these areas have greater diversity? Now, when we say diversity, we can think of it in two different ways. One is diversity in clades, and by that we mean that a clade is a group of related organisms. And if you look at a clade which is about 100 plus million years old, such as birds, you have 10,000 species globally, roughly. On the other hand, you look at a clade that's even older, that evolved before birds did. This is the Tuatara or Sphinodon, and these are 200 million years old and have just one species, or perhaps two, depending on how you define them. But there are many such groups that are as old as birds and have very few species. And there are many others similar to birds that are quite young and have very, very large numbers of species. So we can ask this question in time, saying, why is the diversity of some clades greater? But then if you look at what we call evolutionary radiations, one of the most famous radiations is the radiation of Darwin's finches in the Galapagos. But you have these uh, radiations across many different taxa, from Hawaiian honey creepers to cichlids, African cichlids, to anole lizards, bromeliads, Madagascan vangas, and all of these are actually these very diverse groups, but they're also confined in space, right? So, in a way, we can think about this diversity that we see in nature as the sum of the patterns that we see across clades, that is, these related groups that we've talked about, as well as what we call biogeographic processes or dispersal. And the study of the evolutionary history of diversity and of diversification tends to be our study of looking at patterns across clades and looking at biogeographic processes. But I wanted to add one more complication to this. If you look at a group of frogs, and for example, the frogs of the Western Ghats, we start finding some additional interesting things that make our life difficult. One of the frogs, the genus Raorchestis, which was earlier known as Philautus, um, and it was known as Philautus when I originally encountered this group. So I have a soft corner, let's say, for its old name. Now, this group contains very a great diversity of species in the Western Ghats itself, more than 50 or 60 species. And many of these look dramatically different from each other. And that makes our life quite easy, right? We're like, oh, we look at them and they're morphologically different, and one is able to conclude that they're different species. But in the Western Ghats itself, there's another group called wrinkled or night frogs or Nyctibatrachus, which are also an ancient group and are very diverse, have 40, 50 species. And most of these guys look remarkably similar to each other. And so that sort of then raises the question for us is how do we study and understand these evolutionary relationships so that we can understand how the processes of diversification, what are the genetic changes that took place, what are the morphological changes that took place, and how does that all result in the diversity that we see today. So the first thing that we do is we start out by studying evolutionary relationships. And really, this is the study of how organisms are related to each other and also leads to the classification of organisms. Now, historically, this was done from the time of some of the earliest naturalists, 400 years ago nearly now, such as John Ray and people that even predated the works of very famous naturalists that we remember, like Linnaeus. Linnaeus formalized the system of classification, but he and many others of his time and before him started this process of actually describing species, defining them, and classifying them. And this was all done on the basis of morphology. As early as the late 1600s, John Ray said that when he was interested in plants, and even then, he had identified the seed as or the reproductive features as a good way to identify different species from each other. So it was quite the work. In fact, if you look at much of the work that was done in the 17th, 18th, and 19th century, you'll find actually that on the basis of morphology itself, a lot of very, very sound, rigorous work was done on evolutionary relationships and classifications, and many of those classifications still hold today. However, with the advent of molecular work, we've begun to find that some of those classifications actually were not what we thought it was. So, for example, historically, and we still teach it, I'm sure most of you have learned it this way. I certainly did. And in fact, if you think of the title of today's talk, I talk about herpetofauna. However, herpetofauna, which comprises amphibians and reptiles, is not an evolutionary group at all. The term reptiles, which we still learn, we learned vertebrates are fishes, amphibians, reptiles, birds, and mammals. The term reptiles is not an evolutionary group at all. The term birds is not an evolutionary group at all. So actually, the classic definition of vertebrates or the classes of vertebrates is evolutionarily wrong. And it is wrong because birds and crocodiles actually form the most closely related group from amongst these. And a whole lot of different work, and this is actually, I have a image from a very old paper, but this is, of course, has been more than adequately established now that birds and crocodiles that birds are really sort of evolved are flying crocodiles. They're all flying. The evolutionary descendants of dinosaurs are the closest thing that we have to that group, and the remaining reptiles are actually more closely related to each other. So there really is no group of amphibians and reptiles, and herpetofauna really isn't a thing unless you choose to include birds in it. And these are insights that we were able to derive from using molecular and genetic data to understand these relationships. Similarly, a group that I was very interested in for the longest time, and it continues to be debated to some degree, about who the closest relatives of turtles are. Now, some of these are a little surprising, but there are others that are even more surprising and provide us insights into how evolution takes place. So, if you think of vultures, for example, for a long time, people thought that vultures were all related to each other. And the evolutionary relationships to your left which suggests that all the vultures were closely related, and thereafter closely related to birds of prey and so on to storks. Turns out, in fact, that New World vultures are more closely related to storks, and Old World vultures are related to birds of prey. And Old World and New World vultures is not really a group. Is not a group of birds that are all more closely related to each other than they are to anybody else, right? So they're actually, in a sense, from different parts of the evolutionary tree. Now, that was a surprise. Now, why do these things surprise us? They mainly surprise us because of convergence. So, unrelated organisms evolve structures, traits, or features that have the same functions, and we have some very, obvious examples, such as, many types of fish and marine mammals, such as dolphins, which of course look very alike. But there's enough about their morphology and the osteology and anatomy, that even in the early days people knew to classify them as mammals, right? But then you can clearly see the convergence in form because of function. Similarly, between ichthyosaurs and puffins, and many numerous examples of convergence that you're familiar with. Now, on the other hand, another evolutionary process is divergence, and divergence can happen with, as in these classic examples with finches, in very short spans of time, due to changes in due to isolation from each other, due to changes in habitat, due to adaptation to certain environments, and so on. So we're dealing with this combination of convergence and divergence when we're interpreting from current the flora and fauna that we see, the evolutionary relationships between these species. And today, our work is strengthened by having access to the genetic signatures. So we're able to look at genetic sequences which give us insights into how long ago these may have diverged from each other, because we look at the amount of mutations that they accumulate along these sequences, particularly in parts of the genome which are not selected, and therefore those mutations are sort of accumulating randomly to some degree. And we use those to infer how long ago they may have separated from each other. And that therefore allows us to infer how these, how species and groups are related to each other, but also the processes of diversification, whether there's convergence or divergence and so on, when we compare it to morphological data. So I have here a set of images of mammals, and I'll tell you what they are. And for those of you who want to try this exercise at home, I'll give you like 30 seconds to try it while I have a sip of tea. I'll tell you what they are. One is a shrew. Two is a kangaroo, obviously. Three is an elephant shrew. Four is an elephant. Five is a Sirenian. So that's those dugongs and manatees, which are very closely related to each other. And sorry, I missed the number. Four is the rock hyrax. You guys must be sitting there saying, "Did he say four was an elephant?" Yeah, four is a hyrax, five is the Sirenian, and six is the elephant, right? Now, if you had to draw a tree of the sort that I showed you before, where the closest relatives are essentially connected to each other on the tree, right? That's basically what the phylogenetic tree is. It's a tree of relationships where the closest species are what are called sister to each other on the tree. So you can draw this in any fashion. Essentially, the question is, which pairs of these are most closely related to each other? Which pairs or triplets or whatever it is? Which are the guys that are like really closely related, right? And which are the guys that are like not related to them at all? Like I said, uh, those of you who don't know the answer, regardless of age, feel free to take a piece of paper out or draw it mentally in your head, or type out the answer in the live chat box. Who are the closest relatives in this group? There's the shrew, the kangaroo, the elephant shrew, the hyrax, the uh, dugong slash manatee and the elephant. In this case, an African elephant.
Sir, I think Hyrax is closely related to elephant, right?
Is that Sandeep? Hello? Okay.
Yes, yes. This is me.
Right, right. Now, this the question is not for you; it's for everybody else.
Yes, answer is coming, and there are a few. They are telling about number four.
Okay. Yes,
Few,
yes, answers.
yeah. They most of the students nowadays, I think they know.
So read out some of the answers for me.
Yes, number four, number three, and a few of them number five.
So, are you saying that three, four, and five are closely related to each other? Is that what you're saying?
Yes.
That's not far off the mark, but you would be um, you would have to include elephants in that group. So
Okay.
the really interesting thing in this and there've been many such sort of like uh, you know insights from molecular data, but when this came out about 20 years ago to begin with, people had known for a long time that hyraxes and elephants were closely related.
Yes.
However, and they'd also hypothesized that manatees were related to this group, but elephant shrews actually were classified along with shrews and other relatives because they're shrews and they look like shrews, right? And they were just given the name elephant shrews because of their long name. Now it turns out that entire group of elephant shrews and tenrecs and aardvarks was actually really closely related to elephants and hyraxes, and that entire group was given this name Afrotheria. Uh, Similar work at the time suggested that cetaceans, those marine mammals, their most close relatives were artiodactyls, uh, uh, which are ungulates, and like deer and so on. And in fact, the ancestral marine mammals, ancestral cetaceans, evolved in the Tethys Sea as India was approaching the Asian landmass. Uh, and some of the fossils of the ancestral mammals are found in that region, and they're there in India and Pakistan. So a lot of this was, some of this, a lot of evidence comes from fossil data, of course. A lot of it comes from morphological data, but some of the insights that genetics provides us about these evolutionary relationships is really valuable, and allows us to make inferences about who evolved from whom. Now, all of this also, just to give a very broad, quick background, takes place over evolutionary time when, due to continental drift, the continents are also changing positions. So, on the one hand, there's a whole set of environmental drivers of evolution, but there's groups of animals are connected to each other. Let me just play that again. Are connected to each other uh, over time, and then as these continents separate, they get separated from each other, and some of those come into contact again, and all of these play critical roles in the evolution of individual groups as well as the individual faunas of regions. And in fact, a lot of the theory of plate tectonics was supported by the distribution of some of the fossil fauna as well as extant fauna. So the Glossopteris plants. I saw some of the Glossopteris fossils in a few of the museums that I visited in the north for the first time myself very recently. Piped frogs, carabid beetles. The distribution of these, as you can see in those figures, Glossopteris, for example, found in the south part of the continents of South America, Africa, Australia, and India, kind of tells us about that these continents were joined at one time. And similarly, you can see how the distribution of carabid beetles shows that these continents were together at one point in the past. And of course, evidence for the theory of plate tectonics comes from a whole range of other sources as well, from large, predominantly from geology and chemistry. Nevertheless, now, the reason that is particularly relevant to us going forward is that the history of the Indian subcontinent is extremely interesting, as you would have seen in that animation. India starts out as part of Gondwana, with Africa, South America, Australia, and Antarctica, and then slowly starts to separate 100+ million years ago, and then eventually around uh, around 50 million years ago, leading up to about 30 million years ago, starts and completes contact with the Asian landmass, leading to the formation of the Himalayas and the loss of the Tethys Sea. I just put that in there because I've been recently sort of like doing a little bit of research into what we know about paleontology of India, and there's a humongous body of work that even I was unaware of. And there's a lot of dinosaur fossils from India that we generally don't end up even talking about when we talk about the history of the Indian subcontinent. But more about that at another time. So when we're studying biogeography in this context of the movement of these continents, we study largely. We look at this when I'm coming to the evolution of herpetofauna in India. I would ask questions about how did they disperse here, which is essentially the movement of organisms from one location to another, either actively or passively. And the other are these processes of adaptive and non-adaptive radiation, which is the origin of one or many species from pre-existing ones, which I alluded to as well, right? And for peninsular India, many theories have been proposed about how this dispersal could have taken place, one or a combination of them. The evolutionary trees that you see here are intended to allow us to infer whether there was largely a Gondwanan origin. So the species in India are of Gondwanan origin. When India joined the Asian mainland, they all dispersed out of India, or most of the fauna of India is basically out of India and came into India after the collision with the Asian subcontinent. So these, depending on which scenario has occurred, the phylogenetic trees of relationships would look different, and thus these trees allow us to make inferences about what dispersal scenarios may have resulted in the fauna that we see today. There are similarly many hypotheses about the Western Ghats, and we can hypothesize that fauna dispersed out of the Western Ghats or into the Western Ghats through the Eastern Ghats or the Vindhya Satpura range, known as the Satpura hypothesis, or the Aravalli and other ranges as well. And we can use we can base our inferences on these phylogenetic trees that we can build from sampling these extant fauna. Now, my focus of our work over the last 10, 12 years has been 15 years actually has been on the Western Ghats. And I essentially say that a lot of our work has been about two detective stories. And the two detective stories are related to each other. One is called the Lin shortfall, which is a lack of knowledge of the species diversity of a region, that how many species are there. But equally importantly, the Velation shortfall, or a lack of knowledge of where they're found. And these two questions tend to be quite tightly related to each other, because the knowledge of geography often underpins our ability to make inferences about species. Now, what we did in this project over a 10-year period was we went out and sampled the entire Western Ghats, capturing all the different altitudes, the bioclimatic regions, the geological heterogeneity. So we sampled across these massives, which are these elevational ranges that you see on the figure at the bottom, and sampled most of these 14 mountain complexes across all the elevational gradients over a 7-8 year period. And what we found was that we got some amazing inferences on a large number of groups, and I'll just describe some of those results over here. So one of my students, SP Vijay Kumar, focused his work on bush frogs or Philautus, what is called Philautus, it's called Rochester now, and described 8 or 9 new species, but also found some really interesting signatures of how geography plays a major role in speciation. So most of the species were separated from each other from being isolated on different mountain ranges. We also found uh, that the morphological divergence and the genetic divergence didn't always match with each other. So you had closely related species that looked very similar. You had some closely related species that looked quite different, distantly related species that looked similar, and so on. More interestingly, we found that Vijay's work found that the Western Ghats, the southern Western Ghats, may have been a refuge during the Pleistocene glaciations. And the species in the south were more adapted to the canopy. There were more canopy species found over here, fewer open canopy species, whereas in the north, more of the species had adapted to open canopies. One of the side outcomes of this was also demonstrating that these open habitat species, which may have evolved 15, 20 million years ago, which were likely adapted to grasslands, also show the sort of the spread of the grasslands at this particular point of time in our geological history. More recently, we described a deeply divergent frog called Astrobatrachus curiciana, which actually now belongs to its own family, is the only genus in that family and is the only species in that genus that is known today. This is very similar to another very well-known frog called Nasicopatrachus that was discovered about 20 years ago, which also has only two species. And these are both very ancient frogs in the Western Ghats, and they have no close relatives, and they just have 1 or 2 species in the group. Now, this is very interesting because in the same Western Ghats, you have very similar frogs, which I mentioned in the passing. My student Varun Tosekar studied this group, and there are more than 40 plus species in this group. So why do you get these contrasting patterns where one genus has just 1 or 2 species and another genus has 40 or 60 or more species, which have all evolved, in fact, in this. Some of these have evolved, in fact, in a shorter time than those other groups which have been around for longer. The other contrast, which I also made earlier, was that most of the species in these group look similar, whereas a lot of the Raucestus are, they're morphologically divergent. But here again, you find this greater diversity in the southern Western Ghats. When you come to lizards, you find something quite similar. Again, a very high diversity in the southern Western Ghats. But if you look at the groups that I'm going to show you now, you see another interesting pattern. These frog groups that I showed you so far, at least, are largely of Gondwanan origin. Now, the lizard groups show 2 patterns. You have one group of lizards, the lower blue one, which has many species of what used to be called Philautus, but we renamed that genus as Monellosaurus, Samophilus, or these rockagammas that you find all over the peninsular India, including in and around Bangalore. That these are a group that evolved mostly within peninsular India. Then you have a group that actually originates in Asia and then re-invades India. So they disperse into India. So your agamid diversity is a combination of species that diverge here and other species that dispersed into India from Asia and then and subsequently diverged again. And you see this pattern of a group in both of these, the blue circles, which is the vine snakes to the left and pit vipers to the right. You see a group of species that have diverged entirely within peninsular India, many within the Western Ghats itself. So you get different species in different parts of the Western Ghats. And then you get this other group which diverged in Southeast Asia and then has sort of recolonized the subcontinent, some up to the northeast and others further into peninsular India as well. But these are not the only groups that you see these really fascinating patterns in. And I won't talk too much about any of these, but there's similar patterns that you see in Western Ghats birds, such as the Shulachiles, which used to be called shortwings. you see these similar patterns of divergence. So in a lot of in the work that where I don't explicitly name the species, but both pit vipers and in vine snakes, uh, there was one species that was there's several species in the Western Ghats, but there's widespread species in the case of pit vipers. One of them is Trimerizurus malabaricus. In the other, in the case of Ahethula nazuta, and we find actually that across different barriers of the Western Ghats, such as the Palghat Gap and the Shenkota Gap, you actually get different species. And you would think, oh, I expect that for snakes or frogs because maybe they don't move that much. But you actually find similar patterns in birds, that across these gaps you get different populations and even different species. Uh, and uh, Surprisingly, you even see genetic divergences in mammals, and I think this has been shown for other mammals as well. But even with really large mammals such as elephants, you find these geographical barriers such as the Palgar Gap playing these really important roles. So to summarize what we've learned about the herpetofauna of peninsular India, the work in our lab, and there's widespread work in number of other with number of other research groups as well, including my colleague Professor Praveen Karant at CES. They worked extensively on hemidactylus geckos, as well as other agamid lizards, and some frogs and snakes as well. And a lot of their work also contributes. Their work, in fact, more explicitly looks at the into out of India, out of Asia into India hypotheses, and tries to sort of infer how these two processes may have influenced the fauna that we see today. Uh, Summarizing all of that, herpetofauna of peninsular India have both Gondwanan origins. They're related to the families of the Gondwanan continent, and others have more recent Asian origins and have dispersed into India from Asia. Um, if I had to summarize, I'd say a lot of the families of frogs that you find in the Western Ghats have many of them have Gondwanan origins. A few of them have Asian origins. On the other hand, predominantly snakes and lizards as well. Many of them appear to have Asian origins, but some of them also show. Many of them have diversified extensively in peninsular India, and so some groups include representatives of both in situ diversification as well as into India dispersal. Um, these examples that I repeated again, they're still fascinating, and we still don't know what explains it. Why do some of these show these adaptive, non-adaptive radiations into dozens of species, while others are just represented by just one or two species? And then, of course, there are these many ancient taxa, such as Astrobatrachus that I mentioned, Nasicopatrachus that was described a couple of decades ago. We described an ancient vine snake, which is actually an ancestor to the entire vine snake group from the southern Western Ghats. There's a what's going on with the southern Western Ghats? Why are there so many sort of these ancestral taxa over there? Um, and one of the answers to that, of course, is that or one of the hypotheses is that it was a Pleistocene refugium, and so many species survived there that may perhaps their relatives did not survive in other locations. Anyway, so all of this we've learned a lot in the last 15, 20 years, thanks to research across many groups across India. But these leave many more questions.
Uh, I think Kartik sir is having some internet issues. Okay, let me check. It is okay from my side, but having some problem from. Uh, Let me check, please. I'll be right back. And thank you, all the participants, for asking so many questions. I am trying to connect to Kartik sir so that your questions are answered. So please have patience. I'll be right back. Yes, sir. I think you got disconnected, right?
Yes.
Yes.
Did that just happen?
Okay, okay, okay. No, not an issue. Yes. No problem. Are you presenting, sir? That summary slide?
Oh, when did it get disconnected?
That summary slide.
Oh, okay, okay, okay. Good, good,
Okay.
good. No problem. I wasn't sure when it got disconnected. Let me just try.
Yes, yes, yes.
Screen. You're right.
Yes, yes, yes. Here. Yes, sir.
Okay. What,
Okay.
what, how far had I covered?
That first two or three points.
Oh, okay. Yeah. Well, okay. No, I mean, I won't go over it again. I mean, I've talked about all of this. I just finished by, I was finishing by saying that, the southern Western Ghats is a really seems to be a really fascinating place because there are many sort of ancient taxa, such as the Astrobatrachus, the Nasicobatrachus, and this ancestral vine snake Prohetula that we discovered, the ancestral genus to other the agamid lizards, Microornis. So there's something very cool going on over there. But all of this, in a sense, what our group and many other research groups have found so far is only sort of scratching the surface, and we still have many questions. We've kind of like we've unearthed the patterns, and now we want to we're still asking questions about what explains the patterns that we see. So there's much remaining work to be done. And I wanted to thank a whole range of PhD students that have worked on frogs, snakes, lizards, birds, and so on. People who worked in the lab, postdocs, collaborators, of course, many field staff. Uh, Obviously, takes a village to produce this, and many other people that have probably not been mentioned as well have contributed to all of this, and the generous support of numerous donor agencies and uh, state support. And that's pretty much it. Thank you. Oh yes, I did want to make a plug for current conservation. The young people out there who want to read or write about conservation, go look up currentconservation.org. I believe we're on Instagram. Well, no, I do follow current conservation on Instagram, just not very regularly. But I will leave that to all of you. That's my spiel.
Yes. Thank you so much, sir, for your insightful deliberations. And now it's time for question answer session. So we have received few questions from the audience. So first, yes, first one is
Mhm.
from Sujata De Chaudhary. How mutation rate is used to quantify the species relatedness or evolutionary clock? This is the question.
Yeah, well, it's echoing a little bit. Is it still echoing?
Yes, yes, sir.
Yeah. Yeah. Well, so this is based on our inference about mutation rates for different genes or different sections of genes, And essentially, genes sort of evolve at different rates, and you can also sort of and that is of course influenced by selection. I'm going to give you a very broad answer to this question, and also as you might know that because of the way that genetic information uh, is translated into protein, into amino acids and protein sequences. Some some base pairs or some of those codon those positions are more likely to retain mutations than others. So all of this is factored in when we build sort of like when we conduct an analysis and build and uh, you know build a timeline or a phylogenetic tree, or when we date a phylogenetic tree. So there are many things that go into the process. The simplest step is how do you infer a relationship. So for inferring a relationship, if I assume that those base pairs have an equal chance of mutating at the simplest possible level, the similarity between two sequences tells me how closely related. It may not tell me how far apart they are in time, but if I have three sequences, I can say these two are more closely related because they have more similarities than the third one, or this has more changes than these two, right? So the sequence homology is sort of my first step towards building that tree. My second step is then to infer timelines based on that, and that's in order to do that, we use a number of sort of inference. A number of parameters are used. A number of steps are used to sort of like fix those dates and the times of those and the dates on those trees. So sometimes we can use fossil calibration points to say, oh, with this particular fossil is this many million years old, and this is this ancestral form. Sometimes biogeographic changes, such as when the Indian subcontinent came into contact with Asia, or when the continents separated, when the Isthmus of Panama was formed to join North and South America. All of these sort of biogeographic, these geological processes can also then be used to help you sort of make inferences about dates and relationships. So I would look at it primarily in two axes. One is this who's related to whom, and then this axis tells you the times at which those divergences occurred.
Okay. Next question is from Rupam Devnath. There are some incidents like frogs eating snakes, which appeared in social media during lockdown. What is the possibility behind this? Actually, it is going reverse in the food chain. Or
They were hungry. I mean, there's no reason a large frog shouldn't eat a small snake. Um, I mean, I
yeah,
know that.
but I don't know. I have also seen a video clip that it is actually happening, but I also don't know the reason.
Yeah, well, I mean, there's no reason that a large frog should not eat a small snake. I mean, frogs are pretty, uh, you know, carnivorous. So
Voracious.
large,
Yes.
yeah, large frogs, bullfrogs, pretty much eat,
Yes.
anything. Yeah.
Yes, and sir, I have a question to ask you that geologically, Western Ghat region was once adjacent to Madagascar, right? And
Uh-huh.
so there must be a similar flora and fauna in these two regions. So, is there any similarity in frog diversity in these two places? Have you studied or planning to study like that?
So yeah, so in the sense that there are relationships at a broad level um, between those genera that you find in Africa, in Madagascar, in Seychelles, and in peninsular India. So
Yes.
the group belong to this sort of group. I think they call natatanurans, and they comprise these genus from all of these genera from all of these regions. Now, the relationship also, in a sense, is that sort of India breaks away fairly, at one particular point, and so those you expect those particular relationships with those with the landmasses with whom it had most recent contact. Now, most directly, I think Some of the classic examples are, the well-known sort of the Nasicobatrachus, and some of these deeper relationships keep changing with, with newer studies, and like more genetic data, um,
Mhm.
and more genomic level data. Some of these, relationships have, in newer studies have been shown to be slightly different from what they were before, but. Uh, and so I'm not, very sure what the latest trees show, but Nasicobatrachus, is closest relatives were believed to be this family of zooglossids that are found in Seychelles, um, and uh, but there's uh, broadly speaking, yes. The answer to that is yes. You do find that these belong to the same sort of evolutionary group compared to a whole range of
Yes.
other taxa that are unrelated to them and are found, in the New World and in Asia and so on.
Sir, there is another question that how the evolutionary relatedness of Western Ghat frogs were done by morphological or molecular dating process.
So the trees that I showed you were all based on molecular data, but we
Molecular
also,
data. Okay.
yeah, but those trees all of the trees that I showed are based on molecular data, and so we basically build trees, the phylogenetic trees, only based on molecular data today. And when I say molecular data, basically it's different kinds of genetic data, either gene sequences, genomic data, and so on. Now, uh, in addition to that, we use morphological data to understand the processes of evolution. So the the phylogenetic tree gives you a tree of relatedness. Now, along that tree of relatedness, how did how else did evolutionary processes act on these frogs? So did why do they look similar to each other? Is it because they underwent, that they're very closely related and occupy similar environments? Was there some kind of morphological stasis? Were there no sort of like drivers for morphological diversification? Or if they're different from each other, what are the processes that drove those? Right. So the morphological data, at one level, remains crucial for our inference about the processes of diversification and radiation and so on. At another level, we still do use morphological, in conventional taxonomy, we still use morphological um, characters to define species. So the process of defining a species today is still very much based on uh, on providing a set of morphological characters that can then that then define that particular species, following the rules of the international rules of zoological nomenclature. Now, the ICZ and the code of nomenclature, but I would add to that, saying that sometimes that does pose a challenge, because you have many things. There's a lot of interest today in cryptic species, and the idea of cryptic species are that they're very similar to each other, but they're evolutionarily quite divergent. So the question that it pops up is, how do I, you know, uh, can I change the process of taxonomy to actually define species that are actually morphologically indistinguishable, but still should be considered as different species?
And sir, there is a three or four questions related to the how venomous glands developed or in evolutionary terms in the frogs and also the aposematic coloration.
Well, I'm not quite sure what the question is, in the sense that I can't answer the
Actually, the
question right now.
frogs which have poison glands, how are those evolving? And is there any vibrancy of their color related to it?
Could
And
be.
there is another question,
Yeah.
sir. Glands of venomous frog, do they genetically evolve?
The question is whether um, so animals develop venom or poison, as the case may be. Venom when it's delivered, poison when it's sort of like uh, in the system and is toxic to another animal, for a variety of reasons. It can be for defense, it can be for predation, and so on. Now, in the case of poison in particular, because it's toxic to another animal, it's often developed for defense. And in many instances, it is in fact the lack of palatability of animals, because they taste bad or are toxic, is in fact signaled, because there's no point. You're not going to survive if a bird or a snake has already eaten you, and then it also dies. What is the point, right? It has to recognize, in a sense. So very often these strong signals are developed, along with um, these such defense mechanisms. Um, I'm not entirely sure what the question about how they evolved is, in the sense that that I'm not sure if that's a mechanistic evolution question, or if it's it's a why question. The why question I just answered in terms of it. It often is for defense. Um, the mechanistic question I won't attempt to answer here.
Okay,
But
sir.
it's a,
One more last question,
these
and
are
that
no,
is
I just want to add that these are expensive. Whether it's venom or poisons, they're expensive. They're often expensive. And my colleague in
yes.
CS, Dr. Kartik Sunagar, actually runs the Vinomics lab, and I'm sure he'd be happy to answer these questions more detailed, provide more details, answers these questions. But he's off as um, we all know, and he's often pointed out, these are expensive chemicals to produce. And so animals
Yes.
that evolve them have to make the trade-off between producing these expensive chemicals and the benefits that they provide. And so they evolve when the benefits outweigh the costs, obviously.
Anything advertising, sir, is expensive, right?
Yeah, absolutely.
Just like a peacock tail.
Absolutely, yes.
Yes, yes. And sir, one more last question: that what is your opinion about diversity of Indian subcontinent? Is that because of separation from Gondwana or evolved later on?
Well, I think the summary of my talk was that it's a combination of the two. We have Gondwanan fauna that has subsequently diversified within the subcontinent, as evidenced in many of our frogs and even some of the other herpetofauna. Many fish, actually, that I didn't talk about, but that there's evidence from those groups. And probably many other groups that are today unstudied, right? Like the many invertebrates that people have not studied, and the probably a rich diversity of those groups that have origins um, in our um, from the time that India was part of Gondwana. But there's also this enormous number of fauna that have and flora that have have dispersed into India from Asia. And so that in a way is what makes India so sort of like interesting, because it's really this incredible melting pot. And something that I was unaware of, like you know, some until in fact a few weeks or months ago, is also like historically a number of major mammalian and other groups have evolved in flora and fauna have evolved in the Indian subcontinent. I mentioned cetaceans, so marine mammals actually evolved on the shores of India. Um, Other groups like perissodactyls, so horses and their relatives, their ancestors are sort of we found fossils in India, uh, and many other such groups. So there is actually not just extant fauna that we provide an interesting melting pot for, but historically over geological time as well, India has actually been quite a fascinating place.
Okay, thank you, sir. Thank you so much for for being here, for your valuable time. And I would like to thank the audience, obviously, for their patience listening. And feedback link is provided in the chat box, so please fill it up. And I would like to thank Ambika and Rupam, two of my students, for assisting me and make this event possible. So thank you so much, all.
Thank you, Sandeep, for inviting me, and thank you, everyone.